It's going to be fun, hopefully, guys, to chat about this today. I'm going to try to keep things pretty loose, and I think that was the intention of Al as well. And I want to preface this with saying that I think I should use the term "I" fairly loosely here. And so I'm going to be talking about work that's largely that of several postdocs in my lab, and now we have some graduate students that are also taking new directions with our collaboration with the Aquarium. And so this is really their work, and they were actually kind enough to put together some slides, which I just kind of amalgamated together. So I may call upon them if there are specifics that escape my attention. So I want to spend a little bit of time talking about the science, of course, and then a secondary or perhaps even primary goal of today's discussion is to emphasize the collaboration that we have, this partnership with the Georgia Aquarium. And it's also one of these initiatives that I think we're trying to grow here at Georgia Tech even further. And for our lab in particular, it's been quite fruitful over the last 3 years now. And we hope that maybe setting the stage here and showing you a little bit of what we've been doing, and then a little bit about what the aquarium can offer might inspire some partnerships with other labs here at Tech and other individuals that could benefit from this amazing resource that sits literally an 18-minute walk away. I have timed it, and we go there repeatedly. We've been going there repeatedly for the last 3 years now, almost every 2 weeks, in a variety of sampling projects. And so the title of today's talk is about something very specific with regards to how do you clean up the water in some of these really large ocean systems. Can we leverage microbial metabolisms, microbial powers, if you will, to do what we'd like to do in terms of manipulating the chemistry, which in this case is largely focused around getting rid of nitrates. But working up to that, I'm also going to give you a little bit of background about what the system looks like in general. In fact, our collaboration started with that very question. So we all know what the big stuff looks like in these systems. What does the microbial component that's floating in the water itself look like? And so we've worked on that as well. I'll spend some time talking about that, and then talk about the waste removal side of things and some of the complexity that we're seeing in that system. So just to back, I have to advance it by, there we go. So again, these are great slides that my postdocs put together, and they've been thinking about these questions for a while now, and so have we. Why study the aquarium as a microcosm for thinking about ecosystem processes and thinking about microbial benefits in particular, right? That's what we're doing effectively, and we consider the ocean, or the aquarium at Georgia Aquarium in particular, to be a fairly accurate representation, at least in terms of some of the same ecosystem processes and surfaces that you might see out there in the natural system. But even if you scale it down to the fish tanks that many of us probably keep, I know I have more than one, then some of the same properties emerge, right? So what are the challenges, and what are the benefits of thinking about aquariums as model systems? Well, these are effectively microcosms, right? Any water column out there in the ocean is going to be replete with microbial life in a natural setting. 10 to the 6 cells, 10 to the 7th cells in certain settings. Same is true in aquarium systems. In fact, the microbial load is even higher. Why is the microbial load higher? Well, the bio load, the waste load imposed upon the system by the residents is higher in most cases compared to the natural environment, right? And so the aquarium filtration industry, the water filtration industry in general, has been around and facing challenges of cleaning up our water for a long time, right? We all haven't always thought about it in the context of aquarium systems. We typically think of water treatment in the context of sewage treatment systems or cleaning up water for larger scale agricultural systems, even now kind of verging into aquaculture. But the same challenges are faced in the aquarium. You have a high bioload interjecting a high amount of wastes into the system. Fecal matter, urine coming out of the residents in that system. And as a consequence, we see particularly large amounts of nitrogenous wastes, right? Things like ammonia, at least to start with. And nitrogenous wastes are potentially harmful to the inhabitants of that system. They are harmful. They can be harmful directly. They can impose toxicity in a variety of ways if the concentrations get too high. They can also be harmful secondarily, right? Other participants in the system, microbial participants, can use those wastes to fuel their own production. That can have cascading effects, for example, in terms of algal outbreaks and then stripping down of oxygen, right? All of those things can be exacerbated in an aquarium system because, the bioload is high. But in order to relieve those pressures, you can think about mechanical and physical processes to clean up the system, right? You can take water out of it and replace it with new water, right? You can physically strip out particulate matter from the system. You can filter, in other words, right? Those types of mechanisms exist out there in nature to some extent as well. Water replacement or dilution is an effective way to clean water in natural systems. The entrainment of particulates in certain settings, coastal marshes, is another way to clean water, right? But by and large, if we're thinking about removing the fundamental source terms, the waste terms, right, we're thinking about some microbial chemical transformation that gets rid of them, right? And we rely on the same terms or the same processes in aquarium systems. Can we leverage microbes that utilize waste products to get them naturally out of the system? Well, how do you do that? You ask the microbe to take the waste from a bioavailable form, something that another participant can use to make more biomass, or and then remove it and then convert it into a form, like a gaseous form that leaves the system easily. And so, to a large extent, that's what we do. We leverage the power of microbes to take things, bioavailable forms of nitrogen, convert them into a gaseous form so that they can leave the system. We'll get more into detail about that in just a second. But we've been thinking about this question, and we've been thinking about the microbes in these systems for a while now. And the presumption that I just kind of put forth to you was that in these microcosms, we might expect that we might expect a few things. We might expect that the microbial load could potentially be much higher than we see in situ, right? Because the biomass in these systems is often elevated compared to in situ. That's a prediction. We could also make the prediction that the microbial community, again, it might be doing something functionally that we'd like it to be doing. It might be doing something functionally similar to what we think it's doing out there in the environment. But it is also reasonable to expect that despite our attempts to make the system as quote-unquote natural as possible, the community might look wildly different than what's out there in nature, right? Just basically, does the aquarium microbiome, an artificial habitat, look like that of the natural environment? That probably scales with the scale of the system, right? But we don't really know that for most systems. And we don't have too many examples to play around with that question, right? Especially at the scale that we're getting towards, where you're actually approaching something that looks like nature, right? And so something that looks like nature is the aquarium's Ocean Voyager exhibit, at least in rough terms, right? It looks like nature in the sense that you have a lot of interacting fish species. It looks like nature in the sense that there's a high amount of water movement. It looks like nature in the sense that there's a diel cycle and light availability, not as intense light as you might experience out there in nature, but there's a diel cycle. So and it certainly looks like nature in terms of the chemical composition of the water. Aquarium does a great job of trying to maintain pH. It maintains the alkalinity. It maintains temperature close to what you would expect to see out there in nature, right? But does the microbial community look like what you would expect? All right. So this is the system that we've asked that question in. The Ocean Voyager. How many of you have seen this system? Most of you. Good. If you haven't, you should spend some time there. It's a lot of fun. You can even go and dive in the tank. 6.3 million gallons is the largest indoor exhibit, seawater exhibit in the world. That is correct? That's correct. Okay. Contains thousands of fish and charismatic megafauna, including their prize taxa. The whale sharks has some very large and equally exciting manta rays, in my opinion, and it even has a sea turtle, right? So it looks like nature in the sense that these are some of the taxa that you find out there in nature. It doesn't really look like nature in the sense that you might find all of these co-occurring together, especially at these high densities, right? But from the outside, it looks close to nature. And the water is extensively filtered. We'll get to some of the components or the mechanistic basis of that filtering in just a second. But this water is entirely self-contained, or this system is entirely self-contained, right? Georgia Aquarium doesn't have the benefit of sitting on a coast, doesn't have the benefit of being able to replace dirty water with clean water on a regular flow-through basis. This is Atlanta tap water that's been treated to make it look like seawater. Salt has been added to it. And the entire water turns over in effectively about an hour cycle, and it's cleaned extensively throughout that hour cycle. All right. It first spills over into a sump system. That water is then passed through a series of mechanical/physical filtration mechanisms, the first of which is just a sand filter. The sand filter removes particulates, but the sand filter also has microbial biomass associated with it that is breaking down or starting to break down some of the ammonia waste using oxygen as a terminal oxidant. Right? It then enters into a series of what are called protein skimmers. This is another way to mechanically remove waste from the system. These are just large cylinders, each one's about 20 feet in height, 6 feet or so in diameter, and they're packed full of bubbles. So they aerate these things, and bubbles go from the bottom to the surface, and organic matter adheres to the bubbles and it collects in a foam at the surface, and that foam is skimmed off at the top. It's the same premise that operates it on the beach when you see sea foam accumulating at the top of waves. That sea foam is often discolored. It's discolored because of organic matter. They have 63 or 60-something of those systems operating in Georgia Aquarium. The water then goes into these sulfur-driven denitrification towers that I'll talk about in just a minute. Right? This is a way to leverage the power of microbial metabolism, hopefully to remove a potentially toxic inorganic nitrogen compound, nitrate, and convert it into compounds like nitrous oxide and N2 gas that can leave the system. I'll get into the details of that in just a second. And then finally, the fourth step of the filtration cycle is passing the water through large ozonation systems. I don't remember exactly how many they have. 15, 16? 16 or so. Each one is probably 30 feet tall. It's probably 20 feet in diameter. The water passes through that slowly. It kind of trickles through that. And the water is exposed to high levels of ozone. So you're trying to effectively oxidize the hell out of the water and kill most of the life, the microbial life. Why do they care so much about that step? Well, getting rid of the microbes, you're also getting rid of particulate matter that can diffract light, absorb light, reduce turbidity, or increase turbidity in the system, and decrease your visibility from the front of the tank back to the back. If that visibility isn't high enough, customers don't like it. The commercial goals of the aquarium are not met. So the filtration system, really pretty impressive. We can talk in more detail about that. Al can probably talk in much more detail about that. I can talk about the denitrification system. So let me just go through quickly some of the initial questions that we had about the water column microbiome. Back to this question, well, again, highly artificial system. Some components look like nature. A lot of other components don't, even though we're utilizing some of the same physical, chemical, biological properties to treat the water. What does the water column microbiome look like? So to get at that question, we started sampling with the aquarium about 3 years ago now. Every couple of weeks, somebody in my lab would go over there, typically Zoe, and collect water from the surface of the tank, filter that water down, collect the microbial biomass, and ask what does that microbial biomass look like in terms of how many microbes are there and what species are present. So that went on for a while. Zoe led that effort. Another great postdoc in my lab, Nastasia, started helping with that once she came into the lab. And then some of that work was handed over to her, and she produced a wonderful paper that asks this question: what does the microbiome look like? How many players are in the system? How dynamic is it? So what are your predictions? What, at least in terms of microbial load, higher or lower than nature? Higher, okay. I was a bit surprised by this, so I'm going to skip over some of the target questions and get right into the data. I already talked about the sampling scheme. We sampled every couple of weeks, went out there, characterized what the community looks like, characterized that question, right, or answered that question through a technique called quantitative PCR, where we looked at how many copies of a gene there are in the system, knowing that each microbe carries that gene. Right? Then we use that as a proxy to ask, what does that microbial biomass look like? And then we also did some things, we don't really need to get into the details here, but we did things called metagenomics, metatranscriptomics, to look at what else is encoded in the genomes of these microbes. What are the tools that they carry in their toolkits, and do they turn them on and off? We probably won't have time to get into those details today. So first off, let me skip to the load question. Do we have that? So I want to answer that first. These are data that are plotting two different things over the course of our sampling period, which was about 14 months, starting in June 2016, going to March 2017. And I'm plotting two different things. One is the average turbidity in the water, in the red dots here. And the other is that count of that gene that I was telling you about that serves as a good proxy for how many microbial cells there are in the system. And what's kind of interesting here is, first off, the water clarity fluctuated quite a bit during this period. In fact, this is why we were brought in to begin with to explore the microbiome in this system. There was a noticeable turbidity spike early on in the sampling period that the aquarium got really worried about. High turbidity, again, low visibility, unhappy customers. And so it didn't last for too long. The consequences or the causes of that turbidity spike, the drivers, are still not known. We don't really know what is driving microbial dynamics in this system. And there are microbial dynamics in this system. So if you look at the counts, our simple question was, can we relate microbial cell counts to the bloom period, right? Can we actually confirm that the decrease in turbidity is due to increased bacterial cells? Increased bacterial cells. And it's true, we don't have too many data points here for the quantitative PCR estimates, but you can see a big spike in bacterial cell counts that coincide with the increased turbidity. Mark?
Frank, would you get that bacterial peak if most of that turbidity was organic flocculants or was phytoplankton or something?
No, you wouldn't. No. So this gene is specific to prokaryotes, so you would not see eukaryotic phytoplankton. You might see cyanobacteria. We did not see cyanobacteria. There are hardly any phototrophs in this system.
That's what I was asking, is whether these were riding along with some others. You're saying no.
No, they're not, right? So we're fairly confident that these blue data points mean that bacteria, and there's maybe some archaea in the system as well, are going up and down. It's really nothing else. And what's that?
We had microscopy to verify that.
Yeah. We have microscopy to confirm that. And back to the phytoplankton question, there really is not a I mentioned before there is a diel cycle in the sense that it's dark at night and there's light during the day. But there's not sufficient light to really support a large phytoplankton community. There are not a significant contribution from phototrophs in the system. But the system is dynamic. So microbes went up at the same time as the turbidity peak, went down again after that, and there were a couple of secondary turbidity peaks after the after the initial one a few months later. We didn't see the same increase in microbial cells at that time. There are some reasons that we can get into that might explain that. But the punchline that I want to make here is, there are a couple of punchlines. First off, the system is fairly dynamic in terms of microbial cell load, right? And the second punchline is that if you actually look at the numbers here, and if you have any reference point to what you see in actual marine systems, the microbial counts are about an order of magnitude lower, not higher, but lower than what you would see out there in nature, right? And again, it has a lot to do with how they filter this water, right? The mechanical, chemical systems that they employ, fairly rigorous, but what's even more rigorous and more likely to disrupt what this community looks like is that terminal ozone treatment, right? They pass a lot of ozone through this water. They kill a lot of microbial cells, which has some potentially interesting consequences in terms of selecting for what this community looks like and what might be some of the interesting tools that they carry in their toolkits to deal with that pressure. So it's a dynamic system. It's not only dynamic in terms of load, but if you start asking questions about what is that load comprised of, it's equally dynamic. And so this is a plot that tries to address that. This, again, don't get too distracted by all of the details here. This is, I'll try to simplify it. This is going from left to right across the sampling period. Each of these bars is a different sampling event where we asked what does the microbial community look like at that time. And the different colors correspond to the proportional representation of different microbial groups. And when I say groups, I primarily mean microbes classified at the level of phylum or order. Right? So big phylogenetic divisions between microbial life. How does that vary? Well, at least that's how it's color-coded here. And that's the resolution that I'm plotting here. We also looked at a much, much finer resolution to ask, can we piece out particular strain variants that are going up and down? But the punchline here, again, the colors aren't super important, but the overall picture of the dynamics, I think, is relatively easy to see. You have large shifts in what a microbial community looks like over a relatively short time span. This is a few weeks. You go from a community that looks to be dominated by one particular player to a community that's completely dominated by a different microbial player over a period of a couple of weeks. Those shifts probably occur much quicker than that, or they potentially could occur much quicker than that. And then that community player stays in the system for a little while, and then something happens and the system shifts. Right? So it's actually a very interesting artificial system for exploring the dynamics of community shifts. Right? Again, this is a system that's highly monitored. They know what the nitrate levels are. They know what the ammonium levels are. They know what the pH is doing all the time. They know what the temperature is doing all the time. They know when food comes into the system. They know what food comes into the system. They know how much food comes into the system. Right? They know the health of the animals. Highly, highly monitored. There is nothing that we can confidently attach as a causal event to some of these fluctuations, right? Which I find very interesting. It means the system is kind of poised at this, close to this tipping point, and we don't really understand what the subtle shifts are that may push it in one direction or another. But it's a highly dynamic system, at least in terms of what the taxonomic richness of the community looks like, which we find really intriguing. And so this is another graph that kind of plots that, but is condensing all of those colors into two colors, with those two colors representing two dominant players in the system. Prior graph represented all of the players. This is the two main ones, and showing their fluctuating abundances over time. And you can look at these correlation type graphs and ask whether or not certain players go up and down together or go in the opposite direction together as potentially indicators of whether or not they're interacting. We don't know if these two are interacting, but we have data to suggest that they could be, or there could be, these types of graphs could be indicative of some replacement event, right? Some subtle functional or some subtle selection for a functional difference that we're simply not able to detect yet. But it's intriguing, and again, it points to the dynamics of these systems. And I'm going to skip over some of the data that we have that speaks to the tools and the toolkits of these microbes. I think it's important and interesting to think about what could be structuring these differences. We don't really know yet. There are some interesting properties encoded in the microbes of these systems, including some properties that might allow the microbes to be dealing effectively well with highly stressful conditions. And so one of the most interesting things that did pop up from our more intensive molecular analysis was the detection of some genes associated with producing molecules that the cells store during periods of stress. It's one of several other adaptations that may be present in the system. We don't need to get into the details here, but a punchline emerging from this is that if you think about the microbial load, if you think about what's encoded on the microbial genomes, and if you also ask, what does that entire microbial look like in comparison to a natural system? What we are concluding here is that it looks highly different, right? There are certain players that resemble what you might expect to find out there in nature. These all look like marine microbes, right? But the system overall, in terms of its dynamics, in terms of its load, and probably in terms of what functional tools are most relevant to the system, looks quite different. And this is what this graph is effectively demonstrating, but at the community compositional level. All of these data points here are from aquarium microbiomes. I think these upper ones here are from the Ocean Voyager. These are from data sets from another large ocean aquarium in Chicago. And then these are data from a real world system that we think is probably most closely, if you will, reflective of the conditions that they're trying to recapture in these artificial systems. And the distance between the data points reflects how similar these different communities are to one another. Aquarium microbiomes cluster largely together, but there's a large spread in terms of one sample's relatedness to one another, even between samples that are collected fairly close to one another in time. These are data from, again, a real-world system, and this system happens to be off the coast of Hawaii and happens to be over seasonal gradients. So periods of time where you get large natural fluctuations in physics and chemistry that you might expect the community to respond to. But yet, over that time, all the samples still kind of look like one another. So these systems, much more dynamic, largely artificial, but yet some of the same players are participants in the system. Right? And it's those players that look like what you see out there in nature that we think we want to utilize and see if we can leverage to do tasks like how do you clean the water, right? So I went through at the beginning a description of what the water does as it goes through all the different filtration steps. I mentioned the sand filters, I mentioned the protein skimming, I mentioned the ozonation, and then the third one that I mentioned in there was this sulfur-driven denitrification system. And so I'm going to spend a few minutes talking about that before we go into the discussion. And this is work that was largely led by a postdoc in my lab, now a postdoc in Mark's lab, a guy named Andrew Burns, again, but with help from a lot of folks in the lab. And if Al was here, it would be very nice to have his opinion and description of the history of the creation of these towers. He, along with a collaborator, Eric Hall, formerly at the Georgia Aquarium, have a patent on these unique systems. The mechanistic basis of these systems has been around for a while, and the basis is this. You have microbes out there in nature that can convert a bioavailable form of nitrogen, in this case inorganic nitrogen in the form of nitrate, so an oxidized nitrogen species, in a stepwise manner to a gaseous form, either N2O or N2, right? So you're taking a nutrient from the system that, if it builds up too high, can be toxic, or if it builds up too high, it can spur other microbial growth or phytoplankton growth that you might not want into the system. So you want to get rid of that, right? How do you do that? You convert it into a gas so it leaves the system, right? There's a lot of microbes that can do this. These are microbes that do it under low oxygen conditions, because what they're doing is they're taking something else, some other food source. If they're organic consuming organisms like we are, we would eat a hamburger, we'd strip the electrons off the organic material in that hamburger and put them onto oxygen as a terminal oxidant. These microbes are doing something different because there's no oxygen, or we were creating conditions where there's no oxygen. Instead of breathing oxygen, they put their electrons onto these oxidized nitrogen species and successively reduce them, right, into gaseous forms that leave the system, right? And so the trick here, and like I said, a lot of microbes do this. Most of them are like us. They eat organic matter. However, there are also microbes that eat inorganic matter and strip the electrons off of that and do this same process, right? And so there's a large spectrum of different inorganic electron donors that can be utilized in this process, chemolithoautotrophy by microbes, right? But one of the most common ones is a reduced sulfur species. There's a lot of microbes that we know out there can strip the electrons off of something like hydrogen sulfide or thiosulfate and then simultaneously conduct this process of denitrification, right? And that's the premise for creating these towers. These are sulfur-driven denitrification towers. And so the aquarium has 8 of these things. Each one is about 15 feet tall. You can see them in the background here. This is Zoe cleaning the water. No, she's not. She's collecting a water sample. And they are distributed between what we're calling 2 different pads. So they're all interconnected. The 2 pads are different, though. They differ primarily in flow rate. It's not abundantly clear to me why the flow rate is different between the 2. The system can be manipulated. Flow can be manipulated in the system. But historically, they've been operating at slightly different flow rates. I'll get to some data that shows that in just a second. And this is what they look like. This is as tall as, like I said, 15 feet. You access them from the top. It's kind of a big porthole that you screw open and open up. And they're packed full of basically 3 different things. Gravel in the form of aragonite. So pea gravel provides a couple of different benefits. It can help stabilize pH in these systems, but moreover, it also provides a microbial attachment point. And also, these things also contain the sulfur source. In this case, the sulfur source is not elemental, it is not hydrogen sulfide. It's not thiosulfate. In other words, it's not the more reduced forms of sulfur. It's elemental sulfur in the form of sulfur prills or pellets. I'll show you a picture of them in just a second. And then the third constituent of what's in these systems is simply the interstitial water, right? All of those are potentially different microbial habitats. And we wanted to understand who's living in each of those habitats. Why do you want to understand that? Well, if you can think about who's there in the system, and if you understand what the system properties are, like nitrate removal efficiency, and we know the nitrate removal efficiency in these systems, and I'll get to that in just a second, then you can start to correlate ideal community structure with nitrate removal rates or ecosystem properties, right? And that was the fundamental goal of what we were trying to do here. Do these communities differ? If so, how? Are they complex or are they not complex? Is it a single participant or is it multiple participants acting together? All of that can inform our understanding of, can we recapitulate the system or come up with some better strategy for priming the system elsewhere to do the task, this task of waste removal, right? So we sampled at these denitrification towers on several occasions in 2016 or 2017 with the goal of trying to sample as many habitats as we could within them, right? So I mentioned the 3 big ones, and we collected samples of all of these, right? The prills, or the pellets, the aragonite structuring agent, and then not the inflow water, but the outflow water. And so each of those big towers has a spigot at the end. You can open that spigot and collect It's not really the interstitial water, but it's the outflow water, right? And we wanted to understand who's in that. And here are some of the data that speak to the physical and chemical conditions over the course of the sampling event. And I want to emphasize that we sampled 2 towers and 2 pads? Is that correct?
Yeah.
2 towers on 2 pads, right? But I mentioned before that the pads differ. And these data are plotting the... I must have cut this off. Do you remember what this one is, Zoe?
Should be oxygen, nitrogen.
Yes. That might be right. This is oxygen, I believe. Is that correct? Yeah. So these are oxygen dynamics over the course of about, what is it? A 4-month period or so. Flanking the 2 main sampling events, which are these solid lines here. And each of those sampling events is kind of destructive in the sense you have to open up these things. You're going to get a lot of oxygen coming into the system. Thus the oxygen spikes. So maybe it's not really important to think about the oxygen here, but I think one of these is oxygen and one of them is pH. Okay. The bigger point here is this graph and this graph. Right? This graph is plotting the flow rate. And I mentioned before that these 2 systems differ in flow rate. Pad 1 has higher flow. Pad 2, water moves through the system at a longer residence time. And they fundamentally also differ in nitrate removal. So this is delta nitrate on the y-axis. These are negative values here, if you see. And what you can see here is that there's, at least averaged over time, a fairly large difference in nitrate removal efficiency, with removal being most efficient in pad 2 where there's low flow. And so if we keep that in mind throughout the system, these fundamental difference in removal efficiency and flow rate, then we can start to make some predictions about which community types associate with the most optimal outcome. And so the first question is, well, what do the communities look like? And so I showed you a plot before that when talking about the water column community that showed a bunch of dots close to one another or far away from another as reflecting the similarity between different microbial communities. We're doing the same here, right? But now we have the data for these systems split up between the sulfur pellets, the water, the interstitial water, the aragonite, and then this is one sample of the ocean water, or the water from the exhibit itself, right? And what you can see here is that there are clearly fundamental differences in community structuring. Again, this is taking data on the proportional representation of different microbial groups and collapsing it down into a single data point, right? So there are fundamental differences in community structuring between the different microhabitats. That's obvious. All the water samples, the interstitial waters, clustered to the exclusion of the sulfur samples, which clustered to the exclusion of the aragonite samples. And then there are also differences, clearly, in pad structuring. Right? Pad 1 samples look very different than pad 2 samples in terms of microbial community composition. This is not entirely unexpected by any means, right? We expect differences in community structure. You might not have expected or necessarily thought that there could be strong differences between pad 2 and pad 1, but it was reasonable based on the nitrogen removal efficiency. And so if we peer a little bit more closely into what are the differences, the taxonomic differences, this is a plot that's doing something similar to what I showed earlier, proportional representations of major taxonomic groups in the different habitats, interstitial water, and then the sulfur pellets, and then this is the one open ocean sample. And these graphs are fun because you can kind of pull out the big participants in the system and ask, well, were the ones that you were expected there? And if so, how much do they contribute to the system? But importantly, are there other players in the system that might be equally involved? And so a couple of key players that we want to point out here. The first is a genus of bacteria called Sulforamonas, shown here in purple. It's a group that's present in most of the samples. It's a little bit more abundant proportionally in the water samples, but it's also present in the sulfur, or attached to the sulfur as well. It's there throughout. The bigger difference, at least in terms of what's differentiating, or what could be differentiating the pad 1 versus pad 2 nitrogen removal efficiency, could be things like thiobacillus. And I say that because this genus here, effectively absent from the pad 1 pellets where removal efficiency was lower, but is upwards of 50% of the community in the pad 2 pellets. Right. It's not the only taxa that fundamentally differs between the two. There are things like the xanthomonids that fundamentally differ as well. But if you were to pull out a major driving signal, thiobacillus might be what you focus on. And for good reason. Thiobacillus genus, species denitrificans, is one of the quintessential sulfur-driven denitrifying microbes out there in nature. But what's equally interesting, at least in my opinion, is that a lot of these other groups, including Sulforamonas, including chromatiales, including thiotrichales of different types, they're also known to be capable of oxidizing reduced sulfur species and potentially of also denitrifying. Right? So these are not communities dominated or exclusively by one participant that we know is the only one likely to do this function. Right? There's much more likely to be a variety of different taxa interacting together. We'll get to some functional data that kind of speaks to that in just a second. But what's equally interesting, at least in my opinion, is thinking about these systems from the context of what's driving diversification in microbial guilds. And I think I focus on that because the Sulforamonas pattern in particular is highly intriguing to me. Actually, these should be switched, right? Yeah. So this text should be down here, this text should be up here, but these data are correct, right? So what I want to draw your attention to, again, these are complex graphs, but I think they're interesting. These are, again, the different sample types on the y-axis, water, sulfur, aragonite. And then for each sample, you have two different bars, right? These smaller bars that don't go all the way to the top, that's the proportional representation of different taxa, of these different taxa in the community. So how much thiobacillus there was as a proportion of total is shown by the bars that don't go all the way to the top. And so you can see them spike, as I showed you before in the pad 2. It's effectively the same data that was on the prior graph. And the same thing is being done here for Sulforamonas. And then the taller bars that go all the way to the top, that's looking just at the taxonomic representation of the thiobacillus group or of the Sulforamonas group, and asking within that genus, how much diversity are we seeing, right? And so the punchline here is that in the thiobacillus community, it might have some amount of diversity. There's a few other colors at the top here, but it's largely a single microbial unit that is participating in the system. Upwards of 90% of the thiobacillus community is one taxon. That's completely opposite from this other participant, Sulforamonas, which can do the same thing functionally, or at least putatively, right? Instead, the diversity is all over the place, right? These are highly diverse communities at the species level within the genus. And then if you peer even farther and ask how many different strains of the species of Sulforamonas are present, you would have to have a bar plot that can account for 150 or 200 different colors. In other words, this is a strikingly different community structuring, diversity structuring between two players that are seemingly functionally synonymous, right? One microbe, 200 microbes, right? Presumably, at least from the kind of crude vantage point, same functional types. It's a really intriguing pattern in my mind. It's entirely unclear to me what's driving that. I think if you were looking for a system to explore what are the functional drivers of niche differentiation in microbes, this is a wonderful system to do that. And so I'm going to just conclude with a couple of different graphs that speak to what I said before, that if you start peering into the tools that these microbes are carrying on their toolkit and ask the question, if you start asking the question of how are those tools being used to delimit different functional niches, you'll see that a lot of the microbes share some of the same tools, but there are some fundamental differences as well, and that's what this graph is speaking to. So here we have 3 different taxa that were pulled out of the system, and then within each of these genera, there's 3 different types or units, and those are genomic units that we were able to pull out and assign to these different genera, but they're different genomes. And so we did this by mining metagenomic data and pulling out genomes, right? And then asking to what extent are the genes that we know are involved in different types of sulfur oxidation and different steps of the denitrification pattern kind of speckled throughout the diversity of those genomes, right? And the punchline here, without getting into too many specifics, is that there appears to be a fair amount of modularity, at least in terms of, in some of these organisms, of which genes are carried and by whom, right? And these are highly modular processes in the sense that one compound is converted by one enzyme into another compound, and that compound can then either leave the cell potentially or stay and be activated upon by another enzyme within that cell, or maybe be a cross-metabolite that some other participant can use. And so we're interested in this idea that cross-feeding is likely something that constrains these microbial consortia to look like a certain type of community. There may be these interactions that are chemical in nature where one participant can carry out the process maybe to the step where you produce nitric oxide, which is fine, but not the downstream steps, right? And so that's what we want to explore next. This idea that these systems are not one participant. However, you might have instances where that one participant is the most dominant and relevant participant, but instead, it's likely much more of a network of interactions, right? And that's the way we want to start thinking about these engineered systems, I think. This is a great place to start for that, because we can get to it, we can sample it over time, and we know the functional outcomes pretty well and what generates them, right? So thinking about these cross-feeding in the context of metabolisms that we care about from a practical sense, something that I think the field of engineering microbes to do a task is just starting to embrace. But again, I want to highlight that this system, this collaboration is a great way to do that. I really don't have too many slides beyond this. We did look at patterns of gene expression to see which is active and which is not. But those are the conclusions. A diverse consortia likely mediates nitrogen removal in Ocean Voyager. Drivers of this diversity structuring, still unclear to me, right? That pattern of 200 strains versus one strain, that's completely intriguing. I don't know what's driving that, right? Why not converge all in the same functional unit, right? Diverse steps of S-based denitrification are partitioned among members. That community variation may underlie differences in nitrate removal rates, right? Both systems remove nitrate. They both work. One just works slightly better than the others, probably because the flow rate leads to different physical, chemical conditions that structure the community slightly different, so you get a cascading effect on removal. And then some members or community states may be the targets to ensure optimum removal. If we can engineer a community to look like the optimal state, all the better. Or if we can take a community from the optimal state and use it as a seed to initiate a system, that's an equally beneficial step, right? These are things that we can start to think about now. All right, so I know I took way too long on that in the discussion part and just kind of went out the window, but happy to answer any questions, or more importantly, the folks that actually did the work can answer the questions.